We learned about photosynthesis in other previous vodcasts and found that the final product of the system was most often glucose, an essential organic molecule for life. The production of this molecule marks the transition into a need for cellular respiration in organisms, a way to make use of glucose. For those organic molecules to be broken down into usable energy, it must go through several processes, including glycolysis, which is the first step. This is necessary for any organism with mitochondria. They all need energy in order to function. The word glycolysis itself is derived from two Greek words. It means the breakdown of something sweet or to split the sugar. Gly comes from the origins of organic molecule, and lysis means breakdown, like we've learned in hydrolysis with water, etc. And glycolysis begins in the cytoplasm, actually. It doesn't occur in the mitochondria itself. And the overall importance of glycolysis is to remember that it has the formation of 2 3-carbon pyruvate molecules at the end from a 6-carbon glucose molecule. So that's more with tracing the carbons like we did in the Calvin cycle. It's important to remember how many are at the beginning, how many are at the end. So that'll help you through the process. And we also need to remember that there is a formation of a net of 2 molecules of ATP. And then after glycolysis is finished, the pyruvate is prepped and then used in respiration in the Krebs cycle. And glycolysis is most often aerobic, which means it's oxygen dependent, though other vodcasts will discuss what happens in glycolysis if oxygen is not readily usable. And here are a couple of my images. Here's some basic stuff about reactants and end products, just simplified, and here's another simplified equation for glycolysis with the glucose and then the 2 ATP going in, and then the 2 those pyruvic acid means those 2 pyruvate molecules, and then there's 4 ATP coming out, meaning a net of 2 ATP. And then here's kind of the simplified image of glycolysis, very simplified. We'll go more into detail later about that, more detailed mechanisms, but here are the ATP going in. We generally know what ATP is at this point. The cell's money, so to speak, and it's adenosine triphosphate, the stored energy. And then there's NADH. I can't say the name. It's not going to happen. But it's a coenzyme, and it also helps with the reaction and is a product.
All right. So we first start out with the energy requiring steps of glycolysis with 1 molecule of glucose. This goes through a series of steps where 2 molecules of ATP are used to add 2 phosphate groups and slightly alter the structure of glucose to become fructose. So we get fructose biphosphate. This is then split into 2 pGal molecules, each with 1 phosphate group. Now we're in the energy releasing steps. So some NAD plus comes through and grabs 2 electrons and 1 hydrogen for each of the pGals, producing NADH as a netted product. This in turn also attaches another phosphate group for each. Now ADP comes along and sucks up one of these phosphate groups and produces ATP, one each. So this creates 2 total ATP, which balances out the 2 that were already used in the energy requiring steps. Now it goes through a series of steps to reshuffle the phosphorus end so that it's on the 2nd carbon. Also, you see that H2O is released. This is where on each one a hydroxyl group and a hydrogen are released off of the phosphoglycerates to form PEP. From these PEPs, ADP comes along and takes up that phosphorus to leave behind just pyruvate. And this gives us 2 more total ATP, which is our netted 2 ATP that Nina talked about earlier.
This brings us to the prep step of glycolysis, the formation of acetyl-CoA. This is a biochemical molecule that is produced in the 2nd step of aerobic respiration. It is produced by decarboxylation of pyruvate in the mitochondria. The purpose of this molecule is to carry carbon atoms into the Krebs cycle to be oxidized for energy production. So what happens is one carbon atom is stripped from each pyruvate. The remaining fragment binds with coenzyme A in forming acetyl-CoA. NAD plus picks up the hydrogen and electrons, forming one NADH. Following, and that wraps up our portion of glycolysis and leads us into the Krebs cycle. Here's a video to help you remember glycolysis.
Hey, sugar. Come on over here, sweetie. Let's break it down. Glycolysis is a series of reactions, enzymatic actions, energy transactions. Takes glucose, a molecule so sugary, breaks it down for NADH and ATP. It's an anaerobic cytoplasmic pathway that amazes. Organized easily into 3 phases: investment, cleavage, and energy harvest. Tell me later which one you like best. Investment, activation, energy supplied. Cleavage, our 6 carbon sugar divides. Harvest, we get our energy yield. So beautiful, so intricate, keep your eyes peeled. Glycolysis, come on sugar for the breakdown. For the breakdown. Investments, like striking a match. That energy you put in makes the fire catch. For glycolysis, investments, 2 ATPs, which act as activation energy. Enzymes take phosphates from ATPs, jam them on a glucose, rearranging it to fructose, leaving fructose 1,6-bisphosphate on the table with 2 phosphates. It's highly unstable, moving us to the second phase, the cleaving of fructose bisphosphate, because glycolysis means splitting sugar, you can see. Cleavage yields 2 molecules with carbons 3. One is glyceraldehyde 3-phosphate G3P. It continues on our pathway broken down for energy, but the second one, an enzyme, will immediately convert into a second G3P. Glycolysis. Come on sugar for the breakdown. For the breakdown. Phase 3, G3P gets rearranged and oxidized by an enzymatic assembly line that harvests energy from each G3P, 1 NADH and 2 ATPs. Double this yield for G3P to 2 NADH and 4 ATPs. That's the gross yield for every glucose in. A generous accounting of glycolysis' win, but 2 ATPs were invested in phase 1, so you net just 2. You can use to jump or run, put 2 in, get 4 out, your net gain is 2 ATPs that you can use. If that doesn't seem like very much, it's cause it ain't. There's tons of energy left in pyruvate. The 2,3 carbon molecules we're left with at the end, and what happens to pyruvate's going to depend on the metabolic pathway where pyruvate gets sent. If it's anaerobic, it'll be fermented, but in aerobic cells, pyruvate's termination will be the Krebs cycle and total oxidation. Glycolysis. Come on sugar for the breakdown. For the breakdown. Glycolysis starts with investment of 2 ATPs to the glucose that we started with. The product is cleaved into 2 G3Ps from which the cell harvests NADH and ATPs. This anaerobic pathway is respiration's first phase, billions of years old, evolved in ancient days before O2 accumulated in the seas. Before eukaryotic cells arrived on the scene, it's everywhere, ubiquitous in every organism. Bacteria, sequoia tree, no matter your metabolism, happens in the cytoplasm, doesn't need no organelles. You want to find glycolysis? Look in any cell. Glycolysis. Come on sugar for the breakdown. For the breakdown. Glycolysis. Come on sugar for the breakdown.